Okay, let's unpack this. We've received a fascinating stack of source material, essentially a master guide to molecular diagnostics. Our deep dive today is into the, well, dizzying array of techniques, from foundational PCR all the way to the whole genome sequencing revolution that pathologists and clinicians use every single day. Our mission is twofold, really. First, we need to decode the technology, understand how these tools actually work. But maybe more importantly, we need to understand that crucial decision-making process, the indications. When does a doctor decide, okay, this patient needs this specific genetic test out of all the options?
And that burden of choice, as the sources put it, that's really the central challenge in modern pathology. Before any technique can even be selected, the diagnostician, they have to precisely define what they're looking for. So we absolutely need to begin by establishing two critical concepts: constitutional versus somatic markers.
Right. And this distinction, that basically dictates everything that follows, like the sample type, even the test sensitivity.
Exactly. It's fundamental. If we're looking for a constitutional marker, that means the genetic alteration. It's present in virtually every cell in the body. The classic example here is a single gene disorder like cystic fibrosis, CF,
Yeah.
right, caused by a mutation in the CFTR gene. If you have that mutation, it's in your blood, your skin, your hair. It's part of your foundational blueprint.
Yeah.
So for these constitutional markers, a simple peripheral blood draw or even just a cheek swab is usually perfectly fine for testing.
Okay.
Now, contrast that with somatic markers. These are restricted. They're not inherited. They're actually acquired during life, and they're confined to specific tissues or lesions.
Like cancer.
Cancer is the perfect example. A mutation in, say, the RAS oncogene or that BCR-ABL fusion gene you see in leukemia, that's restricted only to the tumor cells or the malignant cell line. So therefore, the choice of sample immediately shifts. You can't use blood for that. We need a tissue biopsy, maybe a surgical specimen, or sometimes a specialized fluid collection from the site.
Got it. So sample first, then step 2 must be ensuring the test itself is, well, meaningful and reliable, clinically speaking. What are the key metrics a pathologist is looking at before they decide to use a new diagnostic tool?
Oh, reliability is paramount, absolutely. Pathologists, they have to guarantee several core quality metrics. There's sensitivity, how often the test is positive when the disease is actually there. Then specificity, how often it's negative when the disease isn't there.
Right.
Overall accuracy, of course. And critically, reproducibility. Can another lab, using the same method, run the same sample and get the exact same answer? That's crucial for consistency.
Makes sense.
And then, beyond those technical metrics, you hit the practical reality of running a lab. There's the cost, reagents, instruments. How labor-intensive is the process? Is the equipment reliable? And often, maybe the most important thing for the clinician waiting for the result is the turnaround time.
Yeah, you can't wait weeks sometimes.
Exactly. A life-saving diagnosis that's needed today simply cannot wait 3 weeks for some complex assay to run its course.
Okay, here's where it seems to get really complicated, though, and where the source material really digs in. The choice of test, it has to perfectly match the spectrum of genetic anomalies you might expect in that specific patient or population.
Yes, precisely. Because genetic anomalies, they aren't just one thing. They're a massive spectrum. We're talking about everything from, the substitution of a single DNA-based pair,
Tiny changes.
all the way up to the loss or gain of an entire chromosome, or maybe a really complex translocation where bits of chromosomes swap places. A test designed to spot one type of error might be completely blind to another.
And the cystic fibrosis testing example you mentioned earlier, that really illustrates this diagnostic dilemma perfectly, doesn't it?
It really does. Standardized CF panels, they look for a set of the most common CFTR mutations. Now, if you apply this standard test to a patient who happens to be of Ashkenazi Jewish descent, the sensitivity is fantastic. It finds the mutation something like 94% of the time.
Wow, that's high.
It is. But now take that exact same test panel, the same technology, and apply it to a patient population from Asia. The sensitivity just plummets. It identifies less than 50% of the affected patients.
Less than half. Why such a difference?
Because the specific mutations that are prevalent in one ethnic group simply aren't the common ones in another. The spectrum of mutations varies. So if a clinician doesn't take that population variability into account, they might end up falsely reassuring a patient based on a negative test result when the disease is actually there, caused by a less common mutation for that test.
And the problem doesn't even stop there, does it? The source mentioned that even with a really high clinical suspicion for classic CF, and even if you sequence the entire CFTR gene using standard, state-of-the-art next-generation sequencing, You still fail to find a pathogenic mutation in about 10% of patients.
That's right, that 10% diagnostic gap. It's a real thing. And it exists because even these really sophisticated NGS assays, they can be blind to certain types of genetic lesions, specifically things like large kill-base scaled lesions, chunks of DNA missing, and certain complex gene rearrangements that involve segments of DNA just too large or oddly structured for a standard sequencing run to pick up easily.
So it really underscores the need for communication.
Absolutely essential. The primary care doctor, the genetics specialist, the pathologist in the lab, they all have to collaborate closely to select the optimal strategy. And sometimes that might mean using an older, maybe non-sequencing technology to bridge that specific gap to find that tricky mutation.
Okay, let's pivot now and start categorizing the indications for testing specifically for inherited disorders. These are the thousands of Mendelian disorders, single gene things mostly, that can often be definitively diagnosed with DNA sequencing. The source material breaks this down sort of chronologically by the patient's age when they present.
Correct. The timing of when you test is crucial, and the risk factors, the reasons for testing, they definitely shift throughout a person's life.
Okay, starting at the very beginning, prenatal testing. When is a fetus actually offered screening for cytogenetic abnormalities like chromosome problems or specific genetic conditions?
The primary indications here, they really revolve around risk assessment. First off, advanced maternal age. We know that significantly increases the risk of aneuploidy having an abnormal number of chromosomes.
Right.
Second, if one of the parents is known to carry what's called a balanced chromosomal rearrangement, where their own chromosomes are rearranged, but they have all the right genetic material so they're healthy, there's a heightened risk that the chromosomes won't segregate properly during egg or sperm formation, leading to aneuploidy in the fetus.
Okay, so parental factors. What else?
We also test if fetal anomalies are actually detected during a routine ultrasound scan, or if the results from routine maternal blood screening tests, the ones looking for biochemical markers, indicate an increased risk for conditions like trisomy 21, which is Down syndrome.
And the big technical revolution in this space has been the shift toward non-invasive methods, hasn't it?
Oh, absolutely transformative. While the traditional methods, amniocentesis, chorionic villus sampling, CVS, and sometimes umbilical cord blood sampling, those remain the definitive diagnostic tools because they get fetal cells.
They carry some risk, though.
They do carry a small risk, but the emergence of non-invasive prenatal diagnostics, or NIPD, has been huge. This uses tiny fragments of fetal DNA, cell-free DNA, that naturally circulate in the mother's bloodstream.
So just a blood draw from the mother.
Exactly. And using high-throughput sequencing on that maternal blood sample, you can screen for the major chromosomal aneuploidies with very high accuracy, and without posing any direct risk to the pregnancy itself. It's become a standard screening tool.
Amazing. Okay, moving forward in time to testing in newborns and children. What are the classic clinical signs that might make a pediatrician suspect a constitutional genetic disease, something the child was born with?
In newborns and young children, the indications are often quite stark. They frequently involve problems in multiple organ systems or significant developmental delays. So we look for things like multiple congenital anomalies, birth defects affecting different parts of the body. That pattern really suggests a complex underlying genetic program has gone wrong somewhere. We also test if there's a strong suspicion of a metabolic syndrome based on symptoms or newborn screening results. Or, critically, if the child presents with unexplained intellectual disability or significant developmental delay.
And specific features can point towards certain conditions.
Yes, sometimes specific physical features might strongly suggest a particular aneuploidy, like the characteristic features of Down syndrome. Or the clinical picture might fit a known monogenic disease, a single gene disorder, which could either be inherited from a parent, familial, or be a brand new mutation that occurred just in that child, de novo.
Okay. Then as patients get older, the focus of genetic testing shifts again towards diseases that tend to manifest later in life.
That's right. Testing in older patients and adults becomes much more focused, often on targeted risk assessment based on personal or family history. A major area here is testing for inherited cancer syndromes,
When would that be triggered?
usually triggered by a really robust family history of certain cancers, or if the patient themselves develops multiple different types of cancer over their lifetime, or if their cancer onset occurs at an unusually young age compared to the general population. Those are red flags for an underlying inherited predisposition.
Makes sense. What else?
We also sometimes see testing for atypically mild versions of monogenic diseases. For example, an attenuated form of cystic fibrosis, where the symptoms were maybe mild enough in childhood that it wasn't picked up, but caused issues later in adulthood.
Interesting.
And critically, there's testing for neurodegenerative disorders, where a genetic cause is known or suspected, things like certain forms of familial Alzheimer's disease or Huntington disease. In these cases, knowing the genetic status can significantly impact future planning, lifestyle choices, and potential preventative care or trial eligibility.
Now, before we move on from inherited conditions to acquired ones, we have to tackle this really interesting outlier the sources mention, gonadal mosaicism. This indication is genuinely surprising, isn't it? It seems to throw a wrench into the standard rules of Mendelian inheritance.
It is very challenging, both for clinicians to explain and often devastating for the families involved. Okay, so normally, if you have two perfectly healthy parents who have a child with a clear autosomal dominant disorder, let's use osteogenesis imperfecta, brittle bone disease, as an example, we usually assume it was a spontaneous new mutation in that single egg or sperm cell that formed the child, a de novo event.
Right. And the recurrence risk is low.
Exactly. We typically counsel the family that the risk for them having another affected child is essentially back down to the very low background risk in the general population.
But the sources define gonadal mosaicism as the specific scenario where these phenotypically normal, healthy parents go on to have more than one affected child with the same dominant disorder. That completely changes the risk calculation, doesn't it?
Completely. Because it tells you the mutation wasn't just a one-off event in a single gamete. Instead, the mutation must have occurred post-zygotically, meaning after the sperm and egg fused during one of the parent's own early embryonic development. But crucially, it affected only the progenitor cells destined to form their gonads, their ovaries or testes.
So the cells making up the parent's body, their skin, brain, liver, everything else, are entirely normal. They show no symptoms of the disease themselves.
Precisely. They're phenotypically normal. But a proportion of their gametes, their eggs or sperm, actually carry the mutation. This means the seemingly healthy, unaffected parent can unknowingly transmit the disease-causing allele repeatedly to their children.
Wow.
Yes. And the risk for subsequent children is now significantly increased above the general population risk. It's not necessarily the full 50% you'd see if the parent was fully affected, but it's definitely elevated. Identifying potential gonadal mosaicism is therefore incredibly critical information for accurate genetic counseling and future family planning.
Okay, that's a really important, if rare, scenario. Now let's make that essential shift that the sources outlined. Moving from these inherited constitutional disorders to the realm of acquired diseases, specifically cancer and infectious diseases. Here, molecular testing seems to move beyond just getting a diagnosis, becoming a much more dynamic tool for guiding personalized treatment.
Oh, the clinical utility here is massive, and it really spans the entire continuum of the disease from start to finish. Let's take cancer management first. Molecular testing often starts right at the beginning with diagnosis.
How so?
We look for tumor-specific mutations or characteristic cytogenetic alterations that are essentially diagnostic hallmarks of certain cancers. The classic example is detecting the BCR-ABL fusion gene, the Philadelphia chromosome, which is diagnostic for chronic myeloid leukemia, CML.
Okay, so it confirms the diagnosis. And then from there, it immediately moves into guiding treatment. That's where the personalized medicine revolution really kicks in, isn't it?
That's exactly right. This is therapeutic guidance. Identifying specific genetic alterations in the tumor cells directly dictates the treatment strategy. For instance, confirming that a breast cancer has amplification of the HER2 gene means the patient is likely to benefit from targeted anti-HER2 therapies like Herceptin. Similarly, finding a specific activating mutation in the EGFR gene in a lung cancer patient guides the use of specific targeted drugs called tyrosine kinase inhibitors. So the molecular result isn't just an answer to what is it? It's often a direct guide to how do we treat it effectively.
So, diagnosis, therapy guidance. Then once a patient is actually on that targeted therapy, we use these same molecular tools for monitoring how well it's working.
Yes, absolutely. This often involves using highly sensitive techniques like quantitative PCR or qPCR to detect and measure minimal residual disease, MRD. For that CML patient we mentioned, we track the level of the BCR-ABL fusion gene transcript in their blood or bone marrow over time.
And what does that tell you?
If that level remains very low, or ideally undetectable, it tells us the therapy is working effectively, keeping the leukemia suppressed. But if that level starts to rise again, it signals that the cancer is unfortunately returning, often long before the patient develops any clinical symptoms or before it's visible through other tests. It's an early warning system.
And finally, there's the constant challenge in cancer treatment: resistance. Targeted therapies can be incredibly effective initially, but cancer cells are notorious for evolving, often selecting for secondary mutations that make the drug useless.
Yes, this is a major hurdle. Molecular testing is crucial for detecting those acquired drug-resistant secondary mutations. For example, in lung cancer patients on an EGFR inhibitor, the cancer might develop a new mutation to EGFR, like T790M, that prevents the original drug from binding effectively.
So you test for that resistance mutation.
Exactly. Detecting that specific secondary mutation is critical clinical information. It tells the oncologist that the current therapy is no longer working and allows them to immediately switch the patient to a different second-line treatment that can overcome that specific resistance mechanism. It's about staying one step ahead of the evolving tumor.
That makes perfect sense. Okay, the second major category of acquired disease where molecular testing is vital is infectious disease management. This seems heavily reliant on molecular methods for speed and precision, especially compared to older culture methods.
Absolutely. Molecular methods offer truly definitive diagnosis by rapidly detecting the specific genetic material DNA or RNA of the microorganism itself. This is absolutely indispensable for pathogens that are difficult or slow to grow in culture, or where you need an immediate confirmation for treatment decisions.
Like what kinds of infections?
Think about diagnosing HIV or identifying mycobacteria, the cause of tuberculosis, directly from patient samples. Or when you're trying to diagnose a specific neurotropic virus, like herpes simplex virus, causing encephalitis in the central nervous system, speed and accuracy are critical there. Molecular tests provide that. And beyond individual patient diagnosis, molecular techniques are vital for public health surveillance. We use them routinely now to identify specific genetic alterations in microbes, bacteria, viruses, fungi that are associated with drug resistance.
Ah, so tracking resistance patterns.
Exactly. This allows for tailored antibiotic or antiviral regimens for the patient, but also helps track the spread of resistant strains in the community, which is essential in the ongoing fight against widespread drug failure, like with antibiotic-resistant bacteria.
And just like in cancer monitoring, you can use molecular tests to see if treatments are working.
Yes, the principle is the same. We use molecular quantification, again, often using qPCR, to determine treatment efficacy by directly assessing the viral load, the amount of virus circulating in conditions like HIV or hepatitis C. Watching those viral loads drop tells you the antiviral medications are successfully suppressing the pathogen.
Okay, so that really sets the stage for the massive why we use these tests. Now let's dive into the how. We heard earlier that molecular diagnostics started relatively small, but those foundational methods, the ones developed back in the late 20th century, they still play crucial targeted roles today, don't they?
Oh, they absolutely do. The original powerhouses of molecular biology, Sanger sequencing, and especially PCR, the polymerase chain reaction, they completely revolutionized diagnostics back then. And they still allow us to manipulate and analyze DNA with incredible precision in specific contexts.
Let's start with PCR, polymerase chain reaction. It's basically the molecular photocopier, the amplification engine, right?
It really is. PCR is remarkable. It allows us to take incredibly tiny amounts of a specific target DNA sequence sometimes, theoretically, as little as just one starting molecule, and exponentially amplify just that specific segment up to 1 billion-fold or even more in just a couple of hours in the lab.
And why is that amplification so important?
It's essential because most of the downstream analysis methods, like sequencing or gel electrophoresis, they need a lot more material to work effectively and give a clear signal. PCR provides that necessary abundance of the target sequence. The only major constraint, really, is that the target DNA segment needs to be relatively short, ideally under maybe 1,000 base pairs for standard PCR.
Okay, so you've amplified your target. Now, the oldest and for a long time the most trusted way to actually read the sequence of that amplified DNA is Sanger sequencing. How does this foundational technique work, and why is it now being largely superseded for many applications?
Sanger sequencing, developed by Fred Sanger back in the 70s, is built on a really clever biochemical trick. You set up a standard DNA synthesis reaction, like replicating DNA in a test tube, but you add in small amounts of special modified building blocks called dideoxy terminators.
Dideoxy. What's
Yeah.
special about them?
Crucially, these terminators, they lack the specific chemical group, the 3' hydroxyl group, that's needed to add the next base in the growing DNA chain. So they act as chain terminators. They're effectively dead-end molecules for DNA synthesis.
Ah,
And each of the 4 types of terminators, one for A, one for T, one for G, one for C, is labeled with a different fluorescent color dye. So during the reaction, when one of these colored terminators gets randomly incorporated into a growing DNA strand instead of a normal base, that particular strand stops elongating right there.
So you get fragments of different lengths.
Exactly. You end up with millions and millions of DNA fragments, representing every possible length from the start point up to where a terminator stopped it. And each fragment is color-coded at its end according to the last base added. We then separate these fragments very precisely by size using a technique called capillary electrophoresis. As the fragments flow past a laser detector, the machine reads the sequence of colors passing by, and that color sequence directly translates to the DNA base sequence.
It sounds incredibly elegant, but you said it's being superseded. Why?
It's accurate, it's still considered the gold standard for validating findings from other methods. But its main limitations are that it's relatively low throughput. You can only sequence one amplified fragment at a time per reaction, and critically, it requires a single, clean, homogeneous starting template.
And that requirement, a single homogeneous template, that's the key difference that really limits Sanger in complex clinical samples, right? Especially things like tumor biopsies, which are inherently messy.
Precisely. Tumors are notoriously heterogeneous. They've got normal healthy cells mixed in, maybe inflammatory cells, and even the cancer cells themselves are often genetically diverse. They're evolving differently. So if a specific mutation you're looking for is only present in, say, 10% or 20% of the cells in your biopsy sample,
Sanger struggles.
Sanger sequencing will really struggle to reliably differentiate that relatively weak mutant signal from the much stronger background noise of the normal DNA sequence that makes up the other 80 or 90% of the sample. It gets drowned out.
So moving beyond trying to sequence the whole fragment, there are these other PCR-based variations that get deployed when the pathologist knows exactly what single-point mutation they're hunting for, right?
Yes, there are several very clever, highly targeted PCR-based methods.
Yeah.
One of the most sensitive, especially for detecting low-level mutations, is called single-base primer extension. Let's say you're looking for a very specific mutation known to occur at just one single nucleotide position. The classic example is the BRAF V600E mutation, which is highly relevant for diagnosing and treating melanoma.
Okay, you don't need the whole gene sequence then.
Exactly. You don't need to sequence the whole gene or even the whole exon. Instead, you design a PCR primer that sets or anneals just one single base upstream of that target nucleotide site.
Right next to it.
Right next to it. And then you perform a specialized extension reaction using only fluorescently colored terminator nucleotides that correspond to the possible bases at that one position, say, one color for the normal base A and another color for the expected mutant base, T. The reaction only incorporates that single discriminating base.
And then you measure the colors.
You just measure the ratio of the fluorescence from the two colors incorporated. So maybe you see 90% normal blue fluorescence and 10% mutant yellow fluorescence. That instantly tells you the percentage of mutant DNA present in the original sample at that specific spot. It's exceptionally sensitive for detecting low-level mutations, down to maybe 1 of 5% sometimes. But the enormous trade-off is you only get information about that one single base pair, no other sequence data.
Very focused. Okay, another simple, highly targeted, kind of old school technique mentioned is restriction fragment length analysis, or RFLA. What dictates when you might use this method?
RFLA is, well, it's elegant in its simplicity, but its use is quite limited. It's only applicable if the specific mutation you're interested in just happens, by chance, to either create a new recognition site or destroy an existing recognition site for a particular restriction enzyme.
Restriction enzymes being those molecular scissors that cut DNA at specific sequences?
Exactly. Those endonucleases that recognize and cut DNA only at very defined short sequences. So if the mutation changes the sequence right at one of these cutting sites, the enzyme will cut the mutant DNA differently. Maybe it cuts the normal DNA but not the mutant, or vice versa.
And that creates different sized fragments.
Precisely. When you amplify the region around the potential cut site using PCR, and then digest that PCR product with a specific restriction enzyme, and finally run the resulting fragments out on an electrophoresis gel, the normal DNA and the mutant DNA will produce fragments of different sizes, which are usually very easily distinguished on the gel. It's cheap, it's fast, but again, it's entirely dependent on the mutation, luckily falling exactly within a specific restriction enzyme's recognition site, which honestly isn't that common.
Right, a bit niche. Now, what about diseases that are caused not by single base changes, but by changes in the length of a DNA segment, like large dilutions or maybe more commonly expansions of repetitive sequences?
For those, we rely heavily on straightforward amplicon length analysis. This is absolutely critical for assessing a whole class of disorders called trinucleotide repeat expansion disorders. The classic example here is Fragile X syndrome, or FXS. FXS is caused by a massive expansion of a specific 3-base repeat sequence, CGG, within the promoter region of the FMR1 gene on the X chromosome. So, to test for this, we use standard PCR primers that flank or sit on either side of the CGG repeat region.
And the size tells you the answer.
Pretty much. In a normal individual, the PCR product will be a certain size. But in someone with Fragile X syndrome, because of that huge insertion of hundreds or even thousands of extra CGG repeats, the PCR product containing that expanded region will be significantly larger. The size difference between a normal gene product and a product from a patient with a disease-causing expansion is usually quite dramatic, so they are easily separated and sized on a gel or using capillary electrophoresis.
Now here's where we have to bring back one of those older technologies again. The source material specifically highlights that the standard PCR approach for amplicon length analysis can actually fail if the repeat expansion gets too large, like in the case of the full FXS mutation, for example.
That's absolutely right, and it's a really important practical point. If the CGG repeat expansion in FMR1 hits the full mutation range, often meaning hundreds or thousands of repeats, the DNA polymerase enzyme used in the PCR reaction often stalls or completely falls off when it tries to copy across such a long, difficult, repetitive sequence. The reaction essentially fails, leaving you with little or no PCR product from the expanded allele.
So the standard test might look falsely normal or be uninterpretable.
It could, or you might only see the product from the normal allele if the person is heterozygous. So in those specific cases where you suspect a full mutation but the PCR is ambiguous or fails, the pathologist has to revert back to the much more labor-intensive older technique of Southern blot analysis.
Southern blotting. That involves cutting the patient's entire genomic DNA.
Yes. You take the patient's total genomic DNA, cut it up with specific restriction enzymes that flank the region of interest, separate those large fragments by size on a gel, transfer them to a membrane, and then use a labeled DNA probe that specifically binds to the FMR1 gene sequence to visualize the size of the fragment containing the repeat region. It's cumbersome, but it can detect those massive expansions that PCR misses. It's a powerful demonstration that sometimes no technology is truly obsolete if it solves a clinical problem that newer methods cannot reliably address.
A great example. Okay, finally, under the PCR umbrella, let's cover real-time PCR, or qPCR. This method seems essential not just for detecting something, but for actually quantifying how much of it is there.
Exactly. That's the key difference. Unlike standard or endpoint PCR, where you just look at the product at the very end of the reaction, qPCR uses fluorescent dyes or probes to detect and quantify the amount of target nucleic acid sequence being amplified during the exponential amplification phase, cycle by cycle, in real time.
And the main use case is monitoring.
The major clinical application is definitely monitoring chronic conditions over time. This is precisely how we track the efficacy of therapy in many situations. We use qPCR to quantify the level of residual cancer cells, like measuring the BCR-ABL fusion transcript level in CML patients to monitor response to treatment. Or, in infectious disease, we use it routinely to measure the infectious viral load, like the number of HIV or hepatitis C virus copies per milliliter of blood, to see if the antiviral medication is successfully suppressing the pathogen over time. It gives you a number, a quantity, which is incredibly valuable clinically.
Okay, that covers PCR and its many variations for looking at relatively small sequence changes or length alterations. But what do we do when the genetic lesion is enormous? We're talking about things involving hundreds of thousands or even millions of base pairs, huge dilutions, duplications, or rearrangements involving big chunks of chromosomes. PCR and Sanger sequencing are essentially blind to these massive structural alterations, right?
Completely blind. For looking at those large genomic scale alterations, we need to move to different technological approaches, primarily visualization and hybridization methods. And the foundational technique here, which bridges the gap between traditional chromosome analysis, karyotyping, and molecular methods, is fluorescence in situ hybridization, or FAE.
FAIsh. I know it uses much larger probes than PCR primers. Can you walk us through the basic mechanism and explain why it's still so critically important in diagnostics?
Sure. FAIsh uses relatively large DNA probes, often spanning maybe 100 to 200 kilobases or even larger. These probes are chemically labeled with bright fluorescent dyes, different colors for different probes. We then take these labeled probes and apply them directly to chromosome preparations made from patient cells, usually on a microscope slide.
In situ means in place, right, on the chromosome.
Exactly. The DNA in the chromosomes on the slide is denatured, made single-stranded, and so is the probe. The probe then hybridizes, or binds, only to its specific complementary target sequence on the chromosomes. When you look at the slide under a specialized fluorescent microscope, you can literally visualize the location and, importantly, the number of copies of that specific DNA sequence as bright-colored dots.
So you can count the dots to see deletions or duplications.
Precisely. If you use a probe for a specific chromosome, like chromosome 21, and you see 3 fluorescent signals instead of the normal 2 in each cell, that confirms trisomy 21, Down syndrome. If a probe for a region known to be deleted in a microdeletion syndrome fails to bind, or only binds to one chromosome copy, that confirms the deletion.
What else is it used for?
The clinical utility of FLESH is enormous. It detects those numeric abnormalities, aneuploidy. It can spot subtle microdeletions or microduplications that would be completely invisible on a routine banded karyotype. It's excellent for visualizing complex chromosome translocations, especially confirming specific cancer-associated rearrangements. And, very importantly in cancer diagnostics, it's used to assess gene amplification, for example, using a probe for the HER2 gene to count the number of signals in breast cancer cells to determine eligibility for targeted therapy.
And the speed of FLESH is often a major advantage, especially in oncology, correct?
Absolutely. FISH can provide results relatively quickly, often within hours or a day, especially using probes directly on uncultured interface cells. The source material cites the critical example of acute promyelocytic leukemia, APL. This leukemia has a specific translocation, T1517, involving the PML and RRE genes. Patients with APL need to start a specific, life-saving targeted therapy with all-trans-retinoic acid, or ATRA, immediately. FISH can rapidly confirm the presence of that specific translocation, often much faster than waiting for full karyotyping or other molecular tests, allowing clinicians to start the critical ATRA treatment without delay. That speed can be life-saving.
Okay, but FEH requires that we already have a pretty good idea of which chromosomal region we need to look at, right? You have to pick the right robe. What if we want to take a more global survey of the entire genome, looking for large deletions or duplications without any prior knowledge of where the problem might be?
That's where we need array-based technologies. These allow for a genome-wide assessment of copy number changes. The first generation of this technology was array-based comparative genomic hybridization, or array-CGH. You can think of it almost like a competition experiment happening across the entire genome simultaneously.
A competition between the patient's DNA and a normal reference DNA.
Exactly. You take the patient's total genomic DNA and label it chemically with one fluorescent dye, let's say green. Then you take a normal reference DNA sample from a healthy control and label it with a different fluorescent dye, say red.
Okay.
You mix these two labeled DNA samples together in equal amounts, and then you hybridize them onto a special microarray slide. This slide is spotted with thousands or even millions of short DNA probes that represent sequences spaced out across the entire human genome.
So the patient's DNA and the reference DNA compete to bind to those spots.
Precisely. At any given spot on the array representing a specific location in the genome, the patient's green DNA and the reference red DNA compete for binding to the probes. If the patient has the normal 2 copies of DNA at that location, just like the reference, the red and green signals should balance out, and the computer reads the resulting color as yellow, a mix of red and green.
But if there's a difference?
If the patient has a deletion in that region, meaning they only have 1 copy or 0 copies of the DNA, then the normal red reference DNA, having 2 copies, will outcompete the patient's DNA for binding space at that spot. The spot will therefore skew towards red on the readout. Conversely, if the patient has a duplication, meaning 3 or more copies of that DNA region, their green DNA will outcompete the red reference DNA, and the spot will skew towards green. By analyzing the color ratios across all the spots in the array, the computer can generate a high-resolution map of all the copy number gains, duplications, and losses, deletions, across the patient's entire genome.
That's incredibly powerful. But the next generation of arrays improved on this considerably by adding single nucleotide polymorphism, SMP, genotyping arrays. What critical piece of information do these SMP arrays provide that array CGH alone completely misses?
SNP arrays are definitely superior for clinical diagnostics because they provide 2 layers of information simultaneously. Like array CGH, they use probe intensity to measure copy number variations, CNVs, across the genome. But crucially, the probes on S&P arrays are designed to specifically recognize and differentiate between the two common alternative DNA bases, or alleles, at hundreds of thousands of known single nucleotide polymorphism sites scattered throughout the genome.
So they don't just measure how much DNA is there, they also look at the specific genetic variation within that DNA. They provide zygosity data.
Exactly. Zygosity data tells us whether, at a specific SNP location where two different alleles commonly exist in the population, say an A allele and a G allele, the patient has inherited two different alleles, one A and one G, making them heterozygous, or two identical alleles, two A's or two G's, making them homozygous.
And why is knowing the zygosity so important? How does that help clinically? They can detect a specific type of abnormality called loss of heterozygosity, LOH. Why is spotting LOH so clinically vital?
LOH is incredibly important. It refers to regions of the genome where a person should be heterozygous. They should have inherited two different alleles, one from each parent, but instead they only show evidence of having one type of allele across that region. This means that even though they might have the normal two copies of the DNA in that region, so the copy number looks normal on CGH, both of those copies came from only one of their parents.
That's called uniparental disomy, or UPD, both copies from one parent.
Precisely, uniparental disomy. And SNP arrays are the primary tool for detecting these large regions of LOH that indicate UPD. This is why SNP arrays are now routinely used as a first-tier test in pediatric genetics, especially for evaluating children with unexplained congenital abnormalities, developmental delay, or autism spectrum disorder, particularly when their standard chromosome karyotype comes back normal.
And it's essential for specific syndromes.
Critically essential. The ability of SNP arrays to detect LOH and infer UPD is the primary way we diagnose certain imprinting disorders like Prader-Willi syndrome and Angelman syndrome. These conditions are often caused by UPD involving chromosome 15, where having two copies from the mother, for Prader-Willi, or two copies from the father, for Angelman, leads to disease, because genes in that region are normally expressed differently depending on which parent they came from. Array CGH would completely miss this because the copy number is normal. SNP arrays catch it because of the LOH.
Okay, really powerful tools for large-scale genomic changes. Let's move now into some highly specialized types of analyses the sources mention. What if the specific gene defect causing a known inherited disorder is completely unknown? Or maybe the gene itself is known, but it's technically just too difficult or expensive to sequence directly with current technology?
In those situations, we sometimes have to fall back on an older indirect approach called polymorphic markers and linkage analysis. It's essentially using genetic clues nearby to track the disease gene without looking at the gene itself.
How does that work?
The core concept relies on the principle of genetic linkage. If two segments of DNA are physically located very close to each other on the same chromosome, they're almost certain to be inherited together as a block through generations. Why? Because the chance of a physical crossover event, called recombination, happening during meiosis, egg and sperm formation, exactly between those two close points is extremely low.
They travel together.
They usually travel together. So instead of looking at the disease gene itself, we examine other nearby DNA sequences that we know are highly variable between people. These are called polymorphic markers. Common examples are microsatellite repeats, which are short sequences like kakaka repeated a variable number of times, or mini-satellite repeats.
And you track these markers in families?
Yes. We analyze these highly variable marker loci, located near the suspected disease gene, across multiple members of a family affected by the disorder. If we can find a specific version or allele of that marker that consistently travels together or co-segregates with the disease phenotype through the family pedigree,
then that marker allele is effectively tagging the disease-causing version of the gene nearby.
Exactly. We can then use that specific marker allele to predict, with high probability, whether other at-risk family members, like unborn children or younger siblings, have inherited the disease-causing version of the gene, even without ever sequencing the actual gene itself.
Where was this used historically, or is it still used?
It was absolutely essential for the familial diagnosis of many complex genetic disorders before large-scale sequencing became feasible. A classic example is autosomal dominant polycystic kidney disease, ADPKD, where the main causative gene, PKD1, has some very tricky sequence features that made it historically very difficult and expensive to sequence comprehensively. Linkage analysis was the workhorse for family testing for years. It's also still foundational for applications like forensic DNA fingerprinting, paternity testing, and also for monitoring engraftment or chimerism after hematopoietic stem cell transplantation.
Fascinating. Okay, let's shift gears slightly to discuss epigenetic alterations. The key point here is that these are heritable chemical modifications to the DNA or its packaging proteins, the chromatin, that regulate gene activity without actually changing the underlying ATCG sequence itself.
That's the crucial distinction. Epigenetics literally means above or on top of genetics. It involves biochemical mechanisms like the addition of methyl groups to DNA bases, particularly to cytosine bases located in specific regions called CPG islands, which are often found near gene promoters. Another key mechanism is the chemical modification like methylation or acetylation of the histone proteins that DNA wraps around.
And what's the general effect of these modifications?
In very simple terms, increased DNA methylation, especially in those promoter CPG islands, typically acts like a molecular mute button. It tends to shut down or decrease the expression of the nearby gene, effectively silencing it without altering its sequence. Histone modifications can have more complex effects, either activating or repressing gene expression, depending on the specific modification and location.
And where is testing for epigenetic alterations clinically crucial?
It's absolutely essential for diagnosing disorders, where the primary problem isn't a mutation in the gene sequence, but rather the abnormal silencing of a perfectly normal gene sequence. The prime example we've touched on is Fragile X syndrome. While caused by a CGG repeat expansion, the disease mechanism involves hypermethylation spreading across the expanded repeat and into the FMR1 gene promoter, which completely shuts down production of the FMR1 protein.
Ah, so the epigenetic change is the direct cause of the protein deficiency.
Correct. It's also central to diagnosing those imprinting disorders we discussed with S&P arrays, like Prader-Willi and Angelman syndromes. These involve abnormal methylation patterns at specific imprinted control regions on chromosome 15, leading to the inappropriate silencing of genes that should be active from either the maternal or paternal copy.
Okay, so since these are chemical changes, not sequence changes, how does a diagnostic lab actually detect them? You can't just sequence the DNA normally, right?
Right, standard sequencing won't tell you if a cytosine base was methylated or not. So we have to use a special chemical pretreatment called sodium bisulfite treatment. You can think of bisulfite as a highly selective chemical eraser for unmethylated cytosines.
How does it work?
When you treat DNA with sodium bisulfite under specific conditions, it chemically converts any unmethylated cytosine base into a uracil base. Uracil isn't normally found in DNA, but during subsequent PCR amplification and sequencing steps, it gets read as if it were a thymine, P.
Okay, so unmethylated C becomes T. What about methylated C?
Crucially, cytosine bases that are chemically modified by methylation are protected from this bisulfite conversion reaction. They remain as cytosine throughout the process. So after you treat the patient's DNA with bisulfite, amplify the region of interest using PCR, and then sequence that PCR product.
You can tell the original methylation status by comparing the sequence to the reference.
Exactly. Any position where you now see a T that should have been a C in the reference sequence must have originally been an unmethylated cytosine. Any position where you still see a C must have originally been a methylated cytosine. It's essentially a chemical conversion trick that allows a standard DNA sequencing machine to read the epigenetic methylation pattern.
Very clever. Okay, finally in this section, let's turn briefly to RNA analysis. We know DNA is the master blueprint and generally more stable. So why would a lab choose to analyze the RNA copy of a gene instead? What are the key applications?
RNA analysis definitely has its place, despite RNA being inherently less stable and trickier to work with than DNA. The most obvious application, of course, is in virology. We use RNA analysis methods like RT-PCR, reverse transcriptase PCR, routinely to detect and quantify RNA viruses, such as HIV and hepatitis C virus, HCV, which have RNA genomes.
Makes sense for RNA viruses. What about in cancer or other genetic diseases?
In cancer diagnostics, RNA analysis is critical for two main reasons. First, for gene expression profiling. By measuring the levels of thousands of different messenger RNAs, mRNA, simultaneously, often using RNA sequencing or microarrays, we can get a snapshot of which specific genes are turned on or off in a tumor compared to normal tissue. This expression signature can help classify tumors into molecular subtypes that predict prognosis or response to therapy. It's part of molecular stratification.
Okay, expression patterns. What's the second reason?
The second, and perhaps even more common and important application in cancer, is for detecting specific chromosomal translocations that result in fusion genes. We keep coming back to the BCR-ABL fusion in CML as the classic example. In the DNA, the BCR gene on chromosome 22 and the ABL gene on chromosome 9 are often separated by enormous stretches of non-coding intron sequences. Trying to amplify across that huge genomic distance using standard DNA-based PCR is often impractical, inefficient, or simply insensitive.
Right, the introns are huge, but because those introns are naturally spliced out during the process of making the mature messenger RNA.
Exactly. The resulting mature mRNA molecule containing the BCR-ABL fusion transcript is much, much smaller and more manageable. So the standard diagnostic approach is to first isolate the RNA from the patient's cells, then use an enzyme called reverse transcriptase to convert that specific fusion mRNA back into a stable complementary DNA, cDNA molecule.
So you make a DNA copy of the RNA fusion message.
Yes. And this cDNA molecule, lacking all those giant introns, is now an ideal compact target for highly sensitive detection and quantification using real-time PCR, ePCR. This RT-qPCR approach targeting the fusion transcript is the cornerstone of both diagnosing CML and, crucially, for monitoring minimal residual disease during therapy by precisely measuring the levels of that fusion transcript over time. It's far more sensitive than trying to detect the translocation at the DNA level in many cases.
That really highlights how choosing the right molecule, DNA or RNA, is key for certain applications. Okay, we have spent considerable time really digging into the trenches, examining all these specific, often targeted diagnostic tools. Now let's look towards the future, which seems overwhelmingly driven by improvements in performance and radical cost reduction. Next generation sequencing,
Yeah.
NGS.
NGS is absolutely the revolutionary force in diagnostics right now. It's not really one single technique, but rather an umbrella term for a variety of massively parallel sequencing strategies, and the impact has been staggering. It has radically reduced both the time and, maybe more importantly, the cost associated with comprehensive genetic analysis. We're now at a point where sequencing an entire human genome, all 3 billion base pairs, can be done for well under $1,000, sometimes in just a couple of days. That's a feat that was completely unimaginable even just a decade or so ago.
And the primary clinical advantage over the traditional Sanger sequencing, aside from the sheer speed and throughput, is NGS's remarkable ability to handle those messy, heterogeneous samples, right? Especially where the mutation might only be present in a tiny fraction of the cells, which is critical in cancer diagnostics.
Exactly right. That's arguably the biggest clinical win for NGS over Sanger in many contexts, particularly oncology. NGS achieves this much higher sensitivity for detecting low-frequency variants because it doesn't rely on analyzing a single bulk average signal from a homogeneous template like Sanger does. Instead, NGS sequences millions or even billions of individual DNA molecules independently and in parallel.
So it gets statistical power from numbers.
Precisely. It builds statistical confidence through sheer depth of coverage. If you sequence a specific location in the genome, say 1,000 times, we call that 1,000x coverage or depth, and maybe 100 of those sequence reads show a specific mutation, while the other 900 show the normal sequence, you can be highly confident that the mutation is genuinely present in about 10% of the cells in your original sample. Sanger sequencing would likely just show the normal sequence in that scenario. NGS can reliably identify mutations present down to 1 to 5% frequency, sometimes even lower with specialized techniques.
Okay, this ability to sequence millions or billions of fragments simultaneously relies on 3 core principles as the sources outline. Let's try to break those down clearly for everyone listening.
Sure. The 3 basic principles common to most major NGS platforms are: first, spatial separation. The process starts by taking the DNA you want to sequence, breaking it into smaller fragments, and then somehow physically isolating these individual DNA molecules from each other onto a solid surface. This might be a specialized flow cell with tiny wells, or maybe beads, or just randomly attaching them to a glass slide. The key is, each starting molecule gets its own little physical space, ensuring they stay put and don't get mixed up during the next steps.
Okay, isolate the molecules.
Second is local amplification. Once these individual molecules are isolated and immobilized, each one is then amplified right there in place using localized PCR cycles or other amplification methods. This creates a small cluster or colony of identical DNA molecules, all derived from that single starting fragment. The purpose of this is to generate enough identical copies in that one spot to produce a signal strong enough to be detected during the sequencing step. You get millions of these distinct amplified clusters spread across the surface.
So millions of separate amplification reactions happening all at once.
Right, which leads directly to the third and final principle, parallel sequencing. Now that you have millions of these amplified clusters, each originating from a single DNA fragment, the actual sequencing reaction happens simultaneously across all of them. Specialized instruments flow sequencing regions over the surface, and highly sensitive cameras capture the fluorescent signals generated as each base, A, T, C, or G, is incorporated into the growing DNA strands within each cluster, cycle after cycle. This generates staggering numbers of relatively short sequence reads, typically anywhere from 50 to maybe 500 base pairs long, depending on the specific platform, all generated in parallel from millions of starting molecules.
Massively parallel indeed. Okay, so with this powerful technology, how is it actually deployed in the clinical world? It seems like the applications range from very focused to incredibly broad.
Yes, the clinical applications of NGS are generally deployed based on the required scope of the analysis, often starting with the most practical and cost-effective approach for the specific clinical question being asked.
And the most common application by far, the sources say, is targeted sequencing, often using what are called gene panels.
That's correct. This is currently the workhorse of clinical NGS. Targeted sequencing involves analyzing only a carefully selected set of genes, maybe just a handful, or perhaps several dozen, or even a few hundred that are known to be relevant to a specific disease or condition. For example, you might have a panel of 50 genes known to cause inherited cardiomyopathies, or a panel of 500 key oncogenes and tumor suppressor genes relevant for profiling solid tumors.
And the goal here is efficiency.
Exactly. The primary goal is to maximize the sequencing depth, the number of times each base within those specific target genes is read while minimizing the overall cost, and importantly, minimizing the sheer volume of data generated and the complexity of the subsequent bioinformatic analysis. You focus your sequencing power only on the regions you really care about for that clinical indication.
And where are these targeted panels most essential right now?
They are absolutely essential now for detailed tumor profiling in oncology. They help guide the selection of targeted therapies and immunotherapies based on the specific mutations found in a patient's tumor. They're also crucial for understanding the mechanisms of drug resistance when a cancer unfortunately recurs. And outside of cancer, many clinical labs are rapidly transitioning their traditional single-gene tests, which used to be done slowly by Sanger sequencing, over to these much faster, higher throughput, and often more cost-effective targeted NGS panels for diagnosing inherited disorders.
Okay, moving up in scope from targeted panels, we have whole exome sequencing, or WES. This is a much broader approach, isn't it?
Yes, WES represents a much more expansive form of targeted sequencing. Instead of focusing on just a few dozen or hundred genes, WES aims to capture and sequence pretty much all the protein encoding regions in the entire genome. These regions, called exons, make up only about 1.5% of the total human genome sequence. But they are where the vast majority, maybe 85% or more, of known disease-causing mutations actually reside.
So you're casting a much wider net.
A much, much wider net. While WES is not yet considered a routine first-line test for most conditions due to cost and complexity, it has been tremendously successful, particularly in the evaluation of complex pediatric cases involving suspected germline disorders where years of specific targeted tests have failed to yield a diagnosis. These are often referred to as diagnostic odysseys. Because WES looks comprehensively at nearly every known coding gene, it dramatically increases the chance of finding that elusive needle in the haystack, the causative mutation, and finally providing an answer for these children and their families.
And then the broadest approach of all, analyzing pretty much the entire genetic instruction set, whole genome sequencing, WGS.
WGS takes it all the way. It aims to sequence essentially the entire genome, including not just the protein-coding exons, but also the introns, the regulatory regions, and all the vast stretches of non-coding DNA in between. Its use in routine medical genetics is currently still quite limited, largely reserved for specific research settings or for those very challenging clinical cases where even WES failed to find an answer, but the clinical suspicion for an underlying genetic cause remains extremely high.
What unique capabilities does WGS offer compared to WES?
For diagnosing inherited disorders, WGS can sometimes identify causative mutations in deep intronic regions or complex regulatory elements that are completely missed by exome sequencing. But perhaps its most significant unique capability, especially relevant in cancer genomics, is its power to detect novel or complex structural rearrangements across the genome. Things like large insertions, deletions, inversions, and especially balanced translocations where pieces of chromosomes swap places without any net gain or loss of material. WES often overlooks these structural variants because its capture probes are primarily focused only on the exons themselves. WGS provides a much more complete picture of genomic structure.
But the sources are quite clear that WGS is definitely not yet the routine gold standard for clinical diagnostics. mainly due to some significant limitations.
That's correct. Currently, the main barriers limiting the routine clinical use of WGS are, first, the significantly higher cost compared to WES or targeted panels, and often a slower turnaround time for getting results back. But arguably, the biggest bottleneck right now is still the immense informatics challenge.
Meaning the data analysis.
Exactly. Interpreting the massive amount of data generated by sequencing an entire genome, billions of data points per person, requires specialized bioinformatic expertise, significant computational resources, and sophisticated analytical pipelines. This makes WGS inherently slower, more complex, and more challenging to report in a clinically meaningful and timely manner compared to the more focused approaches.
Right, that informatics challenge seems like a recurring theme with NGS. Let's dive into that a bit more. These NGS instruments are technological marvels, capable of generating, as you said, hundreds of billions of base pairs of raw sequence data per instrument run, sometimes per day. The real deep dive challenge then becomes making sense of this absolute digital deluge. This requires specialized bioinformatics pipelines.
It absolutely does. The raw output from an NGS sequencer is just millions or billions of short sequence reads. Turning that into clinically actionable information involves several critical bioinformatics steps.
Okay, the first step in this pipeline, the sources say, is alignment. What exactly are we aligning and why?
Alignment is the fundamental first step. It's the process of taking each of those millions of short sequence reads generated by the sequencer and computationally mapping or aligning them to their correct location within a known human reference genome sequence, the standard map of the human genome.
So it's like putting puzzle pieces onto the picture on the box.
That's a great analogy. It puts all the raw data into its proper genomic context. It allows us to see where in the vast genome each short read actually came from. Once all the reads are successfully aligned, the next critical step in the pipeline is variant calling.
Variant calling. That sounds like where you actually find the differences, the mutations. I assume this is where that sequencing depth we talked about becomes paramount.
It's absolutely crucial here. Variant calling is the process of systematically comparing the patient's aligned sequence data against that standard reference genome sequence, base by base, looking for differences. Sequencing depth, which is the number of independent sequence reads that cover a specific location, is key for confidence. Because sequencing isn't perfect, there are always some random errors. But if you see the same difference from the reference sequence confirmed consistently across many, say 50 or 100, independent reads covering that exact same spot, you'd be highly confident that it's a real biological variant in the patient's DNA, not just a random sequencing error. So where there's sufficient reliable evidence confirmed by numerous reads that meet certain quality criteria, a variant call is made by the software.
Okay, so you've called the variants. The next step is annotation and interpretation, right? This sounds like where the biology and clinical meaning come in.
Exactly. A raw variant call just tells you there's a difference at a certain genomic coordinate, like chromosome 7, position 117,199,637, reference is G, patient is A. That's not very helpful on its own. So the annotation step involves adding layers of information to that variant call, things like what gene is it in? What effect is this change predicted to have on the protein sequence? Example, does it cause an amino acid change or create a stop signal? How common is this variant in the general population? Has this specific variant been previously reported in databases as being associated with disease, pathogenic, likely benign, or is its significance simply unknown of VUS?
And interpreting all this can be complex, especially when you find many variants.
Extremely complex, particularly in cancer genomics or whole exome genome sequencing, where you might identify hundreds or even thousands of variants per patient. The clinical laboratory report then needs to filter and interpret these findings, highlighting the potentially pathogenic or clinically actionable variants, and ensuring the report includes a clear, accessible description of the biological and clinical meaning of these variants to help guide the clinician's decision-making. Dealing with those variants of unknown significance, the VUSs, remains a major challenge.
Okay, and finally, the sources mention even more advanced analytic techniques emerging from NGS data, like mutational signature calling. This sounds like it moves beyond looking just for individual point mutations and starts looking for broader patterns.
Yes, this is a really fascinating and powerful area of advanced NGS analysis. Mutational signature calling involves analyzing the entire landscape of mutations found in a sample, typically a tumor genome, and looking for characteristic patterns or signatures in the types and genomic context of those mutations. It turns out that different underlying mutational processes like exposure to specific carcinogens, UV light, tobacco smoke, or defects in specific DNA repair pathways within the cell tend to leave behind distinct, non-random patterns of mutations across the genome.
Like a fingerprint left by the cause of the mutations.
Exactly like a fingerprint. These mutational signatures can often point directly to the biological mechanism that drove the cancer's development, and this can have huge therapeutic implications. For example, identifying specific mutational signatures that confirm a tumor has microsatellite instability, MSI status, which indicates a defect in the DNA mismatch repair pathway, is now critically important.
Why is MSI status so important?
Because we now know that MSI-high tumors, regardless of their tissue type of origin, are often highly responsive to a specific class of immunotherapy drugs called immune checkpoint inhibitors. So being able to call MSI status directly from the NGS data using these signature analyses can directly guide the use of potentially life-saving immunotherapy for those patients. It's moving beyond just finding the mutations to understanding the underlying processes.
That's truly advanced insight. Okay, this has been a fascinating and incredibly comprehensive look at the ever-evolving diagnostic toolkit in molecular pathology. We started way back with those foundational distinctions between constitutional and somatic markers, which really define the burden of choice for pathologists and clinicians.
And we detail the crucial indications for testing across the entire lifespan, from non-invasive prenatal diagnostics, or NIPD, in the prenatal setting, through pediatric evaluations, right up to the more focused later onset disease testing and risk assessment in adults. We also examine the complexities of those important exceptions like gonadal mosaicism and the really dynamic role of molecular testing and monitoring in managing acquired diseases like cancer and infections.
And then we worked methodically through the diagnostic arsenal itself. The specificity you can get with targeted PCR variations like RFLA or single base extension, the rapid visualization power of FISH for large structural changes, the global survey capabilities of cytogenomic SNP arrays, and their unique ability to detect that crucial loss of heterozygosity LOH, and even the chemical ingenuity required for epigenetic detection using bisulfite treatment, all culminating, of course, in the ongoing revolution that is next-generation sequencing.
And the overarching trend here is really undeniable, isn't it? Because NGS technologies generally provide superior performance, higher sensitivity, broader scope, and are rapidly, continually dropping in cost, the future of diagnostics seems clearly pointed towards massively parallel sequencing, eventually replacing, or at least subsuming, virtually all other current genetic diagnostic modalities. We're already seeing NGS being used for applications that used to require separate tests, from sequencing entire genes to detecting aneuploidy via low-pass whole genome sequencing, and potentially even replacing many older RNA analysis methods with much more comprehensive RNA sequencing, or RNA-seq.
And looking even further forward, the promise of NGS and related technologies extends far beyond just diagnosing known genetic disorders. We're seeing really exciting emerging applications, like using NGS to detect tiny amounts of circulating tumor DNA, or ctDNA, that's cell-free DNA shed from tumors into the bloodstream, as a potential non-invasive blood screen for the earliest possible detection of cancer, or using it for much more sensitive monitoring of treatment response than ever before. And there are even newer third-generation or single-molecule sequencing technologies emerging now that might eliminate the need for that PCR amplification step entirely, potentially impacting the clinical laboratory very soon. It's an incredible pace of change. And this inevitably raises a really important, maybe even provocative final thought for you, the learner, to consider as you digest all this material. If, or perhaps when, comprehensive DNA sequencing becomes truly ubiquitous, incredibly fast, and so cheap that we can potentially identify complex genetic risks for nearly any major disease, cancer, heart disease, Alzheimer's, years, or even decades before any symptoms appear. How should we, as individuals and as a society, navigate that incredibly powerful predictive knowledge? What are the ethical and practical implications of knowing that much about your potential future health? When does testing become ethically required versus when should it remain purely an individual's option? Something to definitely mull over.